we try to keep the time. Ah, okay, time difference. Okay. So now I'm very happy to introduce Malcolm Richardson. Malcolm Richardson is going to talk on asthma and COPD. After the first session this morning on the environment, this afternoon we will talk on, we are not the afternoon, but still in this second part of the morning, on diseases. And Malcolm Richardson is, of course, everybody knows Malcolm. He was the former president of ISHAM, the International Society for Medical Mycology. He is now the new president of the British Society for Medical Mycology, and he comes from Manchester with many, many works, many experience. So thank you very much, Malcolm.
Well, thank you very much for inviting me. It's a great pleasure to come back here to Rennes. We were here last year, but I've been charged with talking about asthma and COPD, but of course behind this I have to mention a little bit about exposure and one or two cases where asthma and COPD were implicated in what I'm going to be talking about in terms of diagnosis and treatment. So I come from the Mycology Reference Centre, Manchester, which is a National Health Service reference centre, and also we became an ECMM Centre of Excellence in 2017, but also I still have a teaching affiliation with the University of Manchester, but desperately trying to retire, but not very successfully. Okay, so these are my conflicts of interest, nothing really of any significance or note for this particular meeting and the topic. So, of course, we've heard already, and we're going to hear more about this afternoon, patients do live in mouldy houses. They certainly do in the UK. And so the top left panel here is a mycologist's dream. I could ask you, please, can you identify by looking at the colonies on this ceiling what they are? Well, I can tell you what they are. There's Stachybotrys, there's Aspergillus niger, of course, there's Penicillium, there's Cladosporium, and so on, and so on, and so on. Why? Why do we have this beautiful picture of different fungal species? The reason is, if you look carefully, through that door is the kitchen, and very humid. A lot of cooking and drying clothes inappropriately, and maybe a tumble dryer which was not vented properly, and so on and so on. So this is a vapor trail coming out of the kitchen and, of course, saturating the wallpaper or the ceiling paper, the plasterboard on the ceiling. So we're familiar with Aspergillus fumigatus, and we'll talk a little bit about this in terms of deposition of Aspergillus conidia in the upper airways. Bottom left, we have Alternaria, and we associate this with thunderstorm asthma, and we've had a lot of thunderstorms in the UK recently. Let's see whether or not we do get cases of clinically diagnosed and laboratory diagnosed thunderstorm asthma. Now, this is an interesting picture. This reminds me, the bottom right-hand side, this reminds me of when I was a student in Leeds in the early 1970s, where I did my PhD on Candida. But here we have mold on the wall of this flat, probably, and it says happy birthday. Those of you sitting in the front rows might be able to read this. It says happy birthday, and there are some lovely snow-capped mountains, and in front of these mountains there's a choir singing happy birthday to you. No. But anyways, is this before or after the party? A nice assemblage of beer cans and wine bottles. Anyway, so this is penetrating damp. This is not because of high relative humidity. This is penetrating damp. But I'm sure we'll hear more about this this afternoon in terms of the mycology of the indoor environment and the reasons for this. And of course, dust. We associate dust with, in our experience, investigating, and I probably have investigated over 400 houses now, both in the UK, the United States, and also in the Nordic countries. More dust, the more Aspergillus you find. What is dust? It's a matrix of corneocytes, of hair, clothing fibers, etc., etc. It's a perfect environment, and we know that Aspergillus grows on the keratin of corneocytes from the stratum corneum. So we have Aspergillus fasciculor here and Aspergillus fumigatus, and here we have dust sprinkled onto a slab plate and a nice colony of Aspergillus fumigatus. Right, this was mentioned previously, and obviously this had appeared in the French press or on the French TV. This was a national scandal. in December 2020, this little boy died. He was 2 years old. His parents were from the Sudan. He was born in the UK. And here we have a very brief history. I'm sort of presenting you with this because there was a suggestion of a background of asthma and fungal sensitization, and that's what I'm going to be talking about. So no real significant medical history. He had flu-like symptoms, and he had the stridor, this noisy breathing, no COVID positive contacts. He had difficulties with breathing. He was treated with nebulizers, and there was some partial relief. Finally ended up in an urgent care centre. This is in a city called Rochdale, which is about 20 miles north of Manchester. And intubated, cardiac arrest, running around the glottis and airway edema, and they were unable to resuscitate this little boy. So pediatric postmortems, as we've learned, and I have been involved now in 4 similar cases since this because of heightened awareness within the government and within the medical community and, of course, within the police. So I was asked to go and investigate this house with the police because these become incidents. There's a possibility of a criminal event, but not in this case or the other ones. So he had significant granulomatous tracheobronchitis. We have learned a lot what sort of samples to be sent for investigations, whether it's biomarkers like galactomannan, how to how can the pediatric pathologists get appropriate samples when they do the postmortem. There are some value for the laboratory, all sorts of issues there. So there's a group of people actually within ESMID of forensic microbiology. It's a study group. It's a fascinating group of people if you're interested. All of these cases, there's extensive candida colonization in the upper airways and in the lungs. There was this focal increase in eosinophils, a marker of asthma. And so the question was raised by the pathologist, was this SAF, severe asthma with fungal sensitization? And the pathologist's opinion, there were 2 PM reports, acute exacerbation of an asthma-like airway disease in part or in full due to mold exposure. So here we are. This is not a pleasant environment to live in. I think you would agree with me. We have a cupboard here that was, there's a little flat, 1-bedroom flat. This could be because of water ingress from a flat upstairs, or it could be, of course, just the environment. We have the bathroom here with this level of mold on the wall. And if you look carefully here, the blue arrow points out where the parents were told, oh, just go away, clean the mold off the wall, everything will be fine. So you can see sort of water drips here where this cleaning had taken place. And they're also advised just go away and buy some paint without actually saying using an antifungal paint to try and cover up all of this mess. And this is the kitchen. So the defense lawyers, because I had to go to the inquest, and that was December last year, tried out to make out a case. This was due to lifestyle, nothing to do with defects in the property. So this is one of maybe 200 flats in a big complex of flats within the city of Rochdale. So that's just to set the scene from what I'm going to be talking about. And of course, you'll hear a lot more about this afternoon. I'm not really going to, but this was mainly this is air samples and contact samples of the visible mold, penicillium, Niger, quite a lot of cladosporium, as we can see here, and so on and so on. But note, no aspergillus was found. Now, these are using grab samples, using this type of sampler. This is what we routinely use. You can have a choice of media. You can incorporate your antifungals into, like itraconazole, into one of the sampling heads, and so on. So this was the scope, the profile of molds isolated in that apartment. Now, because of this, so 1 year, well, actually 7 months after the inquest, now we have this new directive from central government. This became a scandal. It was in the press, on the TV. And we now have the formation of this group. So the Office for Health Improvement and Disparities has been charged with a number of tasks to outline the health risks associated with damp and mold in the home, as if we don't know what these are already. identify vulnerable groups. This is all to do with guidance for landlords. So in this particular case, this was a city council who was the landlord. It's really the private sector landlord. So this is all social housing. And how to provide guidance to assess damp and mold-related hazards. We'll hear more about that this afternoon. And what interventions can landlords take to tackle and reduce the risk of damp and mold. These are big, big tasks to address. So let's see how many years it takes before we can answer some of these tasks that people have been charged with to sort. So we're part of this exercise in trying to improve social housing. Okay, so a few years ago, our former head of respiratory medicine, Ashley Woodcock, and people from the Mycology Reference Centre thought they would look into our pillows, a source of molds. So how often do you change your pillow? How often do you wash your pillowcase? Probably very frequently, but how often do you change your pillow? What's inside your pillow? What's your pillow made of? Synthetic materials or down? So they took a lot of pillows, and as you do, you beat the pillow to death with a mat beater, and you analyze what's in the air. We talked about dispersal of fungal conidia, and we'll hear more about that this afternoon. So they found some fumigators. We've heard from Esther about Rhodotorula. This wasn't mentioned, but we have found out that Rhodotorula is a driver of hypersensitivity pneumonitis, and we have isolated Rhodotorula from bagpipes, and we published this bagpipe player's lung, a new source of interstitial lung disease. So that's just significant finding from pillows. So both synthetic and feather pillows. So and look at these other molds, these some common, some not so common molds found in these pillows. So fungal spores. We have large spores, we have small spores, we have alternaria, and so on and so on, different sizes. And of course, it's the size of the spore that dictates the deposition of the spores in the upper airways. So here we have this, this nice graphic here, showing you, depending on the size of the spore, where fungal conidia, like Alternaria, or right down here, very small spores, Aspergillus, where are they deposited in the airways? And after, how quickly does it take for a small spore to reach right down into the alveoli of the lungs? Okay, and then this is a familiar, graph that we show quite often to make a number of points, and this has been adapted by my former colleague David Denning in Manchester. So we have on the left-hand side, we have invasive pulmonary aspergillosis, and then we have in the middle here chronic pulmonary aspergillosis. This really is what we're going to be talking about this morning to a certain extent. And then we have fungal allergy, ABPA, and SAFs. So that's, again, just introducing the whole background of asthma and COPD. So some more work from us in Manchester, looking at the microbiome of the BALs taken from different groups of patients. We can see ABPA, healthy people, and so on, severe asthma and SAFs. And we can see here the distribution of various species. And of course, the green bars denote Aspergillus species complex. That's going to be mainly fumigators, but other species as well. And some work that one of our PhD students from Saudi Arabia has been doing, still to be published, looking at NGS comparison of dust and sputum. So dust from patients with clinically diagnosed CPA, dust from their vacuum cleaners, and we had to get ethics permission for this. It was a hell of a job to get ethics permission for patients to bring in little samples of dust from their vacuum cleaners, but that's the system you work in, and also to analyze sputum samples, because we get regular sputum samples from patients who come to the CPA clinics in Manchester. But I just want to point out the bars here. Aspergillus penicilloides. Now, this was actually mentioned. Is the speaker from the previous session here? She mentioned it in her list of xerophilic molds. Gee. Okay, you did. Now, this is an interesting, because you will not detect Aspergillus penicilloides using regular media. It's a halophile. And so, you miss it. But look, it's here. It's present in dust, and it's present in sputum samples. And when you look at the literature, so there's some details about this. There's a very low water availability, as you showed this morning. Very diverse habits, the Dead Sea, for example, solar salterns, grains, dried fruits, so basically dried environments. But you need 10% salt in your regular media to isolate it. And a long time ago, so now 1978, Rob Sampson and colleagues published on this. It's very common in household dust samples, but you wouldn't know this unless you use the appropriate isolation media. And it's an interesting organism, and we think this is a very powerful allergen. So we're sort of carrying on this work on Aspergillus penicilloides. But this is interesting. It can stimulate the population growth of house dust mite. We're not here to talk about allergy to house dust mite, but there's a very close relationship between Aspergillus and house dust mites. Okay, so here we have the spectrum of allergic diseases that we associate with various fungal spores. We have Alternaria, Aspergillus, Cladosporium, Penicillium, and we have the various clinical manifestations, allergic asthma, rhinitis. There's a massive amount of overlap between these various clinical presentations. We have ABPA or ABPM, allergic bronchopulmonary mycoses. We have this hypersensitive pneumonitis I mentioned a few minutes ago. We have mycotoxicosis, if we believe in the impact of VOCs, volatile organic compounds, on respiratory health, and so on. So a vast range of different types of allergic reactions. So what is fungal asthma? And again, using this review from my former colleague David Denning, defined as the presence of fungal sensitization or fungal allergy in patients with asthma. Sensitization, immune-mediated response to a fungus without evidence of inflammation or tissue damage, and then fungal allergy. This is an immune-mediated response to a fungus, sometimes associated with tissue damage. So was this the background to that little boy dying in that apartment? Then we come on to COPD. This is where we do have big alterations in the lung architecture. Patients are on steroids, frequent hospital admissions. This is a serious disease, so fungal exposure on top of this is bad news, and also there's a link with malnutrition. So the incidence over the years shown here, and this table could go on and on with sort of more and more reports showing that aspergillosis and COPD are very, very closely linked in different groups of patients, hematology patients, patients with AIDS, and so on. So quite large numbers in these various surveys. And from Madrid, okay, an old paper, but here we have COPD patients. 22% of them had invasive aspergillosis. And we can see here now the admissions as a result of aspergillosis and COPD. So this seems to be on the increase. And again, the same study that was carried out in Spain. So fungal allergy then, in terms of management, now we're going to talk about diagnosis and treatment, because that's really what I've been charged to talk about. So we have these aspects of the management of fungal allergy. So avoidance of the fungus, that's impossible. Control of the inflammatory process, that is possible. Improvement of the airway airflow, reducing the amount of mucus and obstruction, trying to reduce the fungal burden, and also, of course, control of bacterial infection on top of this. So what about antibody tests? So there are people in the audience here who are much more expert than I am in terms of understanding and developing and using antibody tests of various sorts than I am, but the size of the circle here denotes the numbers of patients. So ABPA, IgE determinations are very useful, as are IgG determinations, to a much lesser extent CPA. We use antibody testing very effectively because that's one of the stalwarts of diagnosis, in conjunction with sputum culture, for example, and imaging. And then, of course, we have invasive aspergillosis, and that's what we're talking about with COPD patients. Of course, we're all familiar with the Aspergillus galactomannans test, but that's a pan-fungal test, of course. And over the years, and maybe other people can comment on this, there's an ongoing interest in the use of IgG and IgM detection in patients with supposed invasive aspergillosis. So there's a lot of detail here. I'm not going to go through this line by line, but if we look at fumigators-associated asthma, this is controlled asthma. We have elevations in IgE, specific IgE. We have this level of total IgE, and then we have SAFs. So this is severe asthma with fungal sensitization. We get this elevation of IgE, high levels of total IgE, and interestingly enough, though, normal levels of IgG, whatever the normal level is. And then we have ABPA. That's all part of this overall spectrum of allergic diseases, asthma, specific IgE, total IgE, elevated levels of IgG. We're not too sure what the threshold is here, and depending on the method of detection. And then we have an increase in the eosinophil count, and that's what we did see in that little boy from Rochdale in Greater Manchester. And of course, we have these other investigations that are being carried out as well. Okay, so over the years, there's been a lot of work done here in France, even before I started my PhD in Leeds in the early 1970s. A lot of literature, especially the French literature, but then more recently in international journals, we've had all sorts of methods for determining IgG antibody levels, so from precipitins right through to ELISA and beyond. And still to this very day, we do have in-house immunoassays. These are not particularly well standardized, perhaps more in France than in the UK and elsewhere. We have the ImmunoCap method, and here I've given you a range of cutoffs. We tend to use 40 milligrams per litre as a cutoff, but that was really specifically described by Richard Barton from Leeds in the UK in relation to cystic fibrosis patients, but we do use that in our CPA population. And we have, of course, the newer alternatives, and this is the short list. There are about 21 different companies now that make Aspergillus IgG assays, mainly ELISA. And the point here is a variety of antigens employed, from very crude soups, if you like, of cytoplasmic, somatic, and so all antigens, through to recombinant antigens, and I'll just touch upon those very briefly in a few minutes. So slides from my own collection. This was from many, many years ago. Ottoloni double diffusion or immunodiffusion, counter immunoelectrophoresis, and then our own work back in the early 70s using two-dimensional crossed immunoelectrophoresis, very powerful technique, and then right through now to this highly automated system. And this is the system that is used in the UK, at least, in big departments of clinical immunology, and looking at the variety of immunocaps that you can use. It's a very powerful machine. So that's where our samples end up, from patients with CPA and SAFs and allergic aspergillosis. So nicely all summarized here, and the first author, in fact, two, if not more, of the authors of this article that appeared in Medical Mycology a few years ago now, and I understand this may be revised, but a very useful sort of guidance for the use of antibody detection. It's something we should do as a British society as well. So we have now, this is a French company called LD Bio, and I'm sure some of you are familiar with this point of care test. So Liz Hunter was a postdoc that we had, myself and David Denning. We had and published a few papers using this, but this is just a paper looking at the utility of this particular type of technique in the setting of chronic pulmonary aspergillosis. The point is here that pretty good comparison with the immunocap. So where you have the immunocap sample, 40 or greater milligrams per litre, pretty good concordance with the result, the positive test result with this particular device. So this is just a few euros. We've introduced this type of technique into CPA clinics in China, where we've been working recently, and I'm not sure exactly how popular or what the uptake of this particular POCT has been, but in our hands, it's like a screening test, and then if you get a positive, that can be then sent to our Central Department of Clinical Immunology for the immunocap type of investigation. So here we have this paper here. Now we're talking about specific allergens of Aspergillus as markers for genuine sensitization. So here we have a paper describing 23 allergenic proteins, mostly cross-reactive. So this is now focusing on ASPF1. Some details about this. Belongs to the mitogillin family, and so on and so on. There's a lot of detail about this. No homology with any known fungal gene. This is work done by Paul Boyer in our group. It's a very specific allergen for fumigators. Interesting, almost undetectable in house dust. That's an interesting observation from this paper. So there are many more other specific allergen components that have been described, and the utility of using these in very specific immunoassays. And the Japanese also have been working on this for many years. This now gets extremely complicated. You have asthmatic patients with positive IgE antibody to fumigators, and then if you use recombinant antigens, you can come up with, yes, ABPA, for example, is highly likely. If not, if you get a negative result, it's less likely. So this is now really, really drilling down into using very specific allergens from Aspergillus fumigatus. So we end up with treatments, this very recent review in Clinical Respiratory Journal, and 3 slides showing you aspergillosis. Well, first of all, in COPD patients, so we have dyspnea, we have patients who are antibiotic or glucocorticoid resistance, radiology, and then we have a combination of culture, and we've been developing what we call high volume culture in Manchester, where we may plate out 5 milliliters of sputum or a BAL, not a smaller volume as until recently described and advocated in various guidelines. Galactomannan, of course, and also IgG and IgM, using either ELISA or the LD Bio ICT type of POCT, and of course then NGS. What does NGS, does it really help us? Some of you here may have been developing and have experience of NGS. We see NGS at the moment as the same as sputum culture. It gives you a vast variety and profile of molds in that type of specimen. How on earth do you distinguish between what is colonization and what is really, really significant in terms of allergic disease or disease? So then you've got all these combinations, proven IPA at the left-hand side, and then the treatment that's used, a variety of azole drugs, voriconazole, posaconazole. We have, of course, now we have nebulized powder form itraconazole. We now have PC9945, an inhaled, a very potent azole, or occasionally, of course, depending on the host vulnerability, liposomal amphotericin B. And then we have CPA, chronic pulmonary aspergillosis in COPD. Again, it's a problem, and it can then progress into, into invasive disease. Again, similar clinical presentations and various clinical investigations that are carried out, the same spectrum profiling of diagnostic methods, and then we range from nodules right through to chronic necrotizing pulmonary aspergillosis and the various antifungals, or whether or not antifungals are used, or we just observe the patient. And then finally, sensitization in COPD patients. Here, the patients are hyperimmune, breathing difficulties, and again, the spectrum of culture and biomarker type investigations. And then we have the use of corticosteroids, and then itraconazole or voriconazole for quite long treatment periods. So finally, then we have this, and I'm sure this will be mentioned, or this concept will be mentioned this afternoon. Here we have 62 patients, and this is a patient from some of the people we do know here. This is from France. I know one or two people. There could be one or two people in the audience here who are authors of this paper. Anyway, they looked at over 60 patients with COPD. They used an electrostatic dust collector, and of course we may hear more about that this afternoon, an alternative way of collecting dust, instead of pinching the dust from a vacuum cleaner bag. This is using quite a nice, sophisticated way of collecting dust. And in summary then, fumigatus or aspergillus more frequently detected during the exacerbation. High levels of antibodies, where there was a high total mold count in the indoor environment. That's an interesting but not unexpected observation. And aspergillosis was diagnosed in 2 patients who presented with increased levels of antibodies. So this is the conclusion of the paper. Antibody detection, levels of antibody associated with chronic lung alteration, functional alteration, and or domestic mold exposure. Okay, finally then, what about monitoring? And I'm not going to encroach upon anybody else's time, but these are available on Amazon now, 100 euros or more. There are many, many of them, but just note here that we haven't looked into this, mold and VOCs. Don't know if anybody's had any experience of these, but because of this highlighting of this tragedy in Rochdale 2.5 years ago now, there's quite a lot of research money in the UK for investigating mouldy houses. So we have, in collaboration with some inventors and housing surveyors, we are looking at the use of real-time monitoring. So here we have, again, the mouldy bathroom. Now this is a device that could be put inside a house where there are damp problems or there are vulnerable patients. And so a constant stream of air over a whole series of slides, and using photography, the development of these colonies is monitored in real time, say over 48 hours, and then that image is sent to an AI, artificial intelligence type of station, and the colony is identified based on photo recognition of the colonies. So we've also been working on AI recognition of fungal colonies. A lot of people are doing this, but this is just a prototype. It's going to be installed in a number of problem houses in and around the UK. Let's see where we go from this. But as I say, there is grant money now for this type of investigation. So that's me, and vast literature, of course. I found these 2 reviews here. This one in particular, this covers more than just allergic disease and aspergillosis and COPD. It covers all aspects of human exposure to fungus. So thank you very much for your attention. I'm very happy to take any questions.
Could I ask you?
Yeah, so thank you very much, Malcolm. You have a real huge experience. I don't know why you are all specialists on molds in Manchester. Is it a humid weather? Humid climate?
This is why we come to Rennes, because it's always sunny here.
Could I ask you, please, to send that one slide with the government issued?
Yes, you mean in terms of the committees going forward? Yes, of course.
please.
It's in the public domain, but I can send you that slide, of course. There's a big document associated with that summary.
So, yes, some question.
I wanted to take a photo, but it wasn't quick.
Well, thank you again for the talk, and I'm really glad you talked about the case I was just asking before.
Yeah.
But for what I recall also from this case, so this was even a private housing situation.
No.
No?
No.
Okay.
So, but, I mean, housing, it's the same in France. You have owner-occupied dwellings, houses, flats, whatever,
Yep.
mansion houses, a few, and then you have what we call social housing.
Yeah, yeah.
So there are two categories of social housing. The social housing, which is organized by housing associations, so they are, if you like, private landlords. And then you have vast areas of social housing, which are, if you like, the landlord is the council, the city council, but then you have a third group of landlords. So these are, if you buy a house, big house in wherever you come from in France, and you subdivide it into flats, it's a Victorian house, you don't want to spend too much money, so that's a private landlord. And they're the ones who are so resistant to improving the property,
Yep.
they ignore all the complaints. Hence this government directive now, you've got to get your act together because
Yep.
we're going to come after you if you don't. So we have an organisation called Environmental Health Officers, and they can actually declare a house unsuitable for habitation based on what they see. Or the landlord has to fix things, and if they don't, then the consequences are pretty severe,
Yeah.
which is good.
No, I'm really glad you mentioned that because those were my two concerns I wanted to raise. So I've been in the Netherlands now for 2 years, although I moved there and then left and came back. And I remember when I arrived, the two first houses I had were really in poor conditions. And I feel like nowadays that we are moving so much, we are migrating so much, we are not buying suddenly something in a place we are not sure we will stay. In science, we move a lot. So how can we... I mean, I felt at the time, who should I complain to about this situation? That's why I was so interested in knowing if there's regulatory bodies checking the housing conditions, and especially in cities like the one I am now, that buildings are from the 60s still. And another concern, just so if I can quickly mention.
This will be also the debate for this.
Yes.
Okay, no, but this is another concern because you mentioned the AI, and I feel like now everyone is doing AI nowadays. And is it really necessary for this issue? To have
Well,
AI
I mean, I'm just thinking of that particular situation. I mean, the alternative is that the image of this developing colony, let's say it's Cladosporium, is then viewed by a human being to say, yes, this looks like Cladosporium based on the topography and the color of that colony, or you make a slide prep, but this machine will send that image to an photo recognition library, and we have over 50,000 images now of both developing mold colonies and the microscope preparation. So it gives you an answer. I mean, this is very, very familiar to bacteriologists with these automated track systems like the WASP or the Chiestra system. It's all photo recognition, and there's the answer on the screen. So.
Okay, thank you very much. We have to move to the next. You will ask your question after, no problem. So now I'm really happy to welcome Riina Richardson. So Riina is also working at Manchester, and she will talk on a very particular clinical situation, cystic fibrosis. And you are really expert on the field of mold, of course. So thank you very much, Riina.
Okay, thank you so much for inviting. And yes, quite a challenge to talk to this audience about CF and fungal infections, because so many of you are involved. But I decided I will take more of a sort of clinical approach, practical approach, the reality check, how we do and how we think about things, having both clinical and laboratory background myself. My disclosure, sorry, I failed to use the template, but it says it all. And none of this is relevant for the topic of the talk. So first starting, so not everybody is necessarily completely aware what cystic fibrosis is. A couple of facts about it, and facts that are relevant for what we're talking about today. So yes, it's a genetic disease, inherited disease, caused by mutations of the CFTR genes, or cystic fibrosis, transmembrane regulated gene, and the mutation leads to dehydration of fluids in the body. So that is in the respiratory tract, GI tract. Everything becomes sticky and gooey. Saliva is thick and sticky. Everything is concentrated in a way. And that leads to mucus plugging in the lungs, blockage of fluid secretion, inflammation, and tissue damage and fibrosis. We also know that recurrent pulmonary exacerbations that are mainly caused in childhood by bacterial pathogens, first normally starting with Staph aureus as little kids, and then later with Pseudomonas. They are the predominant feature for many patients. Chronic colonization with Candida and Aspergillus has been linked with exacerbations and more sort of rapid decline of lung function. Patients colonized with Aspergillus have an increased risk particularly for APPA. So APPA is the disease in fungal disease typically seen in CF. And the exacerbations are treated with oral and IV antibiotics. Many of them are sort of on suppression with oral antibiotics, and then they come into hospital or via outpatient OPAT care, have sort of pulses of IV antibiotics. I think the other thing that is really important in this context to know that there are sort of completely novel disease-modifying treatments now in clinical practice. So we have the CFTR modulator therapies, which basically partially or quite fully return to normal function, and that stops the sticky, gooey excretions. So they become more or less normal. So the question with CF and fungi is always, what is colonization and was it infection? And as Malcolm's presentation showed clearly, that we have to separate that immune response and having antibodies does not equal to disease. Humans have evolved in rainforests and lived in mud huts through the evolution of humankind, and our immune system and everything has been developed so well that we respond to infection, and some of the response is just a reflection of health. So if antibodies don't automatically mean infection, finding fungus doesn't mean infection. How do you tell the two apart? And it becomes even more difficult, like Malcolm mentioned, NGS. The more sensitive methods are used, the more fungi you find. What on earth does it mean? And that is a big problem clinically. And there's a massive risk for overtreatment and complete loss of antifungal stewardship. And the reality is that the clinical evidence of benefit of antifungal treatment is very, very limited in CF patients beyond APPA. So APPA, yes, we know it works and it's needed and it works. But actual clinical trials showing that somebody is colonized with fungus X, you treat that and the fungus goes away, does the patient get better? That evidence is very shaky. Clinical trials are very limited. The other thing is to remember is that CF itself, the mutation itself, doesn't affect your immune system sort of directly that badly. It's really historically the old-fashioned treatments, steroids and other things. So the question is that what is looking forward? What's going to happen? So we have these novel treatments. They will reduce the need for antibiotics. They will reduce the need for steroids and other immunosuppressants. Biologicals are being used. And then later, of course, for lung transplant. So what is it going to look like in the future is another question. So is the fungus an innocent bystander? Is the question. just a couple of facts. These have been mentioned already. We know there are loads of species. A couple of them only are associated with human disease. This is evolving a little bit with more and more research and NGS and other methodologies. But I think that's sort of the bigger picture. The important thing that has been mentioned was topic of yesterday was about, how do we resist fungal infections? It's not really about us. It's mostly about the weakness of fungi, and that the poor thermotolerance of most fungal species is the thing. Now, how that is relevant in CF setting is that many of them require nebulizers and saline nebulization just to loosen that sticky gooey stuff, to moisturize it so that they can cough things up. So they use a lot of nebulizers, and the systems are often cool. And the nebulizers, they can become dirty and colonized, so they are inhaling who knows what. And so those fungi, so often the sort of oversimplified scenario is that you get a sputum sample from a patient, you grow it in the laboratory at 25, 37 and 45. If it doesn't grow in 37 and 45, it's an environmental contaminant, can't cause human disease, can't live in human body temperature. Now, with this setting, it probably can colonize that nebulizer. So that's not as straightforward as we thought. This is from the Casa de Val review, this picture, but it's just a reminder that our ability to resist fungal diseases has these two main pillars. That's the immune side of the things, and then the endothermy. And I'm not going to cover the endothermy anymore because I think that's what needs to be said about it, really, other than keep your eyes and ears open, what's happening, new pathogens may be popping up. But the immune side, yeah, we're doing all sorts of things, and this is a wonderful picture of how it all works. Yeah. Important thing, every part of the innate and adaptive immunity and all the soluble components of both systems and all the cellular components of both systems, ranging from complement to innate cells of macrophages and neutrophils and to your B and T cells, to your antibodies. The whole cycle, the whole immune system is dealing with it. But this is the curve that Malcolm already briefly showed, but it's really key to think what a fungus can cause in humans. So yeah, if you're an average person with normal immune system, having a few spores in your airways, you're in the middle there. And the curve shows that that's colonization, there's no damage. And the weaker you become, your immunity is defected, the more damage you start seeing because the fungus is able to invade tissues. Important thing, the stronger isn't the better. We always think something a little bit more detergent in the washing liquid, it'll be good. No. Optimal is good, because the stronger you start seeing damage again, but that is now caused by the immune system. Stupid thing, doing too much. So the range of diseases is there. And as I said already, the main disease in CF is in the hypersensitivity side. They are hyperreactive. And that's their type, sort of what is typical. However, the important thing is that CF patients represent the whole spectrum of that in different stages of their life. So they are at risk of invasive aspergillosis if they develop a hematological malignancy. If they need a lung transplant, they are then pushed to the left. And if we manage to control their immunological, the hypersensitivity, the burden of disease with the new wonderful drugs, they are maybe in the center. So the whole spectrum. So there are two components, the sort of physical sticky gooey lung side, But then there is the immune spectrum, which is independent, different genes, and the whole spectrum of humanity is there. So this is I like it, pretty good review recently from Journal of Fungi, actually, yeah, this year, summarizing the most common allergic fungal diseases. And the only place where CF is mentioned is with APPA, allergic bronchopulmonary aspergillosis. So that's just to say, yeah, that's recent review. And that is based on a number of papers. I just picked up a couple of them. And this is now from 2014, so a couple of years back, definitely. And they showed that 15% of CF respiratory specimens were positive for moles, and that was in contrast to 2% of non-CF respiratory specimens, and 32% of CF patients had one or more cultures positive for moles, in contrast to less than 3% in non-CF patients. Yeah, they are more colonized, full stop. Nobody's arguing about that. The interesting part of the paper is this list of species. And you can see that there is only a couple of them, a couple of lines there where you have bigger numbers with 3. The last row can't see anything of the detail, but you can see that it's only 1s and 2s, single digits here, but there are a couple of them with hundreds of cases. And those are Aspergillus species. So Aspergillus fumigatus and flavus. And when you group them together with the type Aspergillus fumigatus and Aspergillus non-fumigatus, Scedosporium, and then what they considered was contaminant, the only group that gives you p-value is the Aspergillus one. So, okay, definitely, CF Aspergillus is important. And this is an older review already, but similarly summarizing all the literature up to the point, saying that yes, Aspergillus fumigatus colonization is bad news, basically, in CF. And then what do you do with antifungals is unclear. So going back to this table now, diagnosis. How do you do the diagnosis? The next one of the columns there, just so that the back row can see. It basically says you have your immunology that you need to do, microbiology, and your radiology. And in immunology, it's IgE mainly, but IgG as well. And specific IgE and G for Aspergillus or other moles, if you suspect other moles, but mainly Aspergillus, as said. And then total IgE, so what proportion it is of that. Eosinophilia is a typical finding. Microbiology, you do culture and you do Aspergillus PCR. And radiology, yeah, you do that anyway, but mainly to rule out other diseases, CPA and such. So we looked at this. We looked at, because we have one of the country's biggest CF centers, the pediatric CF center and then the adult CF center in Manchester. We had decided, we did a lot of workup on the sputum samples from these patients to identify exoviala, to identify all sorts of moles and other things. The question was, what's the point of it? Does it relate to anything? And we looked at particularly at exoviala, because that was one of the main projects that we were working on. And we wanted to see that how did it go pre and post when they first were identified with exoviala? How did their lung function evolve? And you can see from here that the midline is where the identification, the day 0 was, the first exoviala found. There's no real difference left and right. the patients go their own way. We even looked at then that many of them were treated for exophiala, and they may have had co-colonization with Aspergillus or other things. So they got courses of mold-active antifungals. That made no difference. And we looked at all sorts of parameters, all clinical things and so forth. And really, the number of p-values is very limited in some link to CF-mediated diabetes. Don't know what that means. But the conclusion of our paper was that despite the frequent isolation of exophiala, it was not associated with any clinical progression of the disease. It didn't make difference left, right, or center. At the same time, it's really interesting, Paul Verwey and others from Nijmegen coordinated this Dutch study published in Medical Mycology, same year, and they found that basically the summary of the paper was that this study suggests that Scedosporium and exophiala are probably no major pathogens and made no difference. And this was a nationwide study. And these are the kind of being at the clinical interface. Yes, the lab can do wonderful. We can isolate all sorts. We can do NGS. What does it mean? And stay away from treatment unless you have any evidence that it works. But at the same time, we did an analysis. This was just triggered. So we analyzed the beta-D-glucan levels in our CF patients because we got a gut feeling that when they got sick, and we did beta-D-glucans as per the protocols, they were always positive, always positive. So we wanted to analyze, and we have a tissue bank for CF patients because we are a center. So we went to the tissue bank and we collected patients, CF patients, a set of them pre and post and during 6 samples per patient with exacerbations, without exacerbations, and looked at their blood beta-D-glucan levels. And I'm sure we all know what beta-D-glucan is, but it's basically a panfungal sugar. I call it always fungal dandruff being shed all over the place when it's around. And disappears it's cleared by neutrophils taking the liver. Sugars are dealt with in the liver, and that's basically their life cycle. So we noticed that there were some patients who had very high level of beta-D-glucan antigenemia, and it was persistently. You can see on the top graph there that they had over many months, that's a year follow-up, they were all the time over 300 or 200 or hundreds, clearly positive. Others were all the time negative. And when we plotted them against various things, age was a thing that came up as a p-value. And it's interesting that when you're younger, the spectrum is wider. But when you get older, it's low. Our interpretation of that was that it actually probably reflects basically the natural loss of patients through death, and the survivors cannot have continuous antigenemia. So beta-D-glucan is highly immunogenic and causes systemic inflammation. And if you have systemic inflammation for 30, 40, 50 years, you end up with problems. So the older patients can't have that. Either they cope better with BDG and get rid of it, or they die away. And so yes, that's interesting. It's not to be ignored, fungi, but not just an innocent bystander, probably. But but what do we know about treatment? Cochrane, they haven't bothered anymore. They did this in 2014, maybe due to be done again, but they basically didn't find any systematic randomized controlled trials for treatment of APPA in CF, and they basically say we don't know. We could do better, we could do those clinical trials, but nobody's doing. Just a mention about fungal biofilms broadly. So they develop on any surfaces that are either non-renewing, non-shedding, like lines and catheters and whatevers, or nebulizers, spacers, or they develop in cavities, preformed cavities or sinuses. Now, they are innately resistant to treatment, but the key thing in the CF setting is that they are very rarely in human bodies single species. And it's not even single couple of species, but mixed kingdom biofilms. So you have bacterial, fungal biofilms. And the thinking is that they are protection against each other and immunity and antibiotics or antifungals. So should you treat for both to get rid of them? Otherwise, if you treat for the mixed biofilm only with antibiotics, it ends up with with a fungal skin, not penetrating anymore, and you have persistent bacterial infection, or the other way around. So we looked into this as well, already a couple of years back now. And as the title says there, interestingly, the intravenous antibiotics reduce the presence of Aspergillus. And that just reflects that they are very symbiotic. They hang on to each other, they hold hands. And when the other one goes with it. So the assumptions we make and extrapolations we make from other settings have to be made very carefully. It's a different disease entity. And this is an interesting study from a local study from here, and summarizing, very clearly that you get a lot of Aspergillus in these patients. So the environmental exposure is there. So we are back to basic. Yes, Aspergillus is important, and that maybe we should do some surveys, house surveys. And this leads to this final part of my work, which is about Norman's paper from Manchester about our CF unit. So Pierce Ward is our CF unit, and it's a very old-fashioned part of the building where they have a wonderful courtyard with trees and other things inside there, and the CF patients can get fresh air. They open the doors, and so much about ventilation and filtration. So that's the Pierce Ward. And because of some construction work and other reasons, we did weekly air sampling there for about a year, 14 months. And all the sampling data has been put together into this paper for that over a year period. And not going into detail of any of this, but I think the findings are, and then, yes, they were combined with the Met Office data, so the UK basically weather forecast data, and wind speeds, temperatures, rain, no rain, everything was put together into magical statistical analysis. The findings are less surprising. So Aspergillus fumigator spores were more abundant during summer months. Yeah, we have leaves, we have plants, excellent. High counts were driven by higher temperatures and lower wind speeds. Okay, not surprising. But indoor counts directly correlated with outdoor counts. Well, obviously so, if you open windows and doors. But the link of indoor counts with the exacerbations of APPA is so well established. So this comes to the point that what instructions do we give these patients? Do we tell them not to do composting, not to go outside, keep windows closed, and so forth? And final reminder, although we are very clear now what diseases are there, definitely the story isn't all written and clear, so keep your eyes and ears open with climate change. New things may be coming our way. So, summarizing, it's important to remember that CF patients are not one group. They represent a broad spectrum of immunological responses, ranging from deficient to hyperreactive. The main fungal pathogen, as we know at the moment, and probably stands, is Aspergillus fumigatus, and APPA is the most common disease. But getting to the point, and the point of this afternoon is that, of course, we know that prevention is better than cure, but controlling exposure is better than prophylaxis. How do we control the exposure? And that requires better understanding of the environmental exposure sources so that we can implement better non-medical, non-drug-based approaches to keep these patients safer. So at the same time, yes, CF management is evolving, term tolerance is evolving, that's all messing up the picture. So just a sort of reminder that, let's keep our eyes and ears open. That's me, thank you.
Thank you so much, Riina, for this brilliant presentation. So we have time perhaps for one question, but once again, we can talk on this topic this afternoon. So, Joanna. Yeah.
Thanks, Riina. That was really insightful. My question is very sort of philosophical. With the advent of the CFTR corrector drugs being approved, how do you see the landscape of fungal diseases changing?
It requires monitoring. Who knows? But the simplified thinking is that there is nothing wrong with the immune system of these patients other than if we give them steroids and biologicals and whatevers. But essentially, there's nothing wrong. And if we can get rid of the sticky mucus by fixing the problem, they're just like anybody else. But I don't know.
How do you get rid of that?
With the correcting drugs. Yeah, so the regulated drugs, they are amazing. When you look at these patients, they are amazing.
Yeah, I think we will have results in a few months or a few years because everybody is monitoring that, and it's really impressive. So
it is.
Yes.
It is. Yeah.
Okay, so thank you very much. And now I'm very happy to welcome Françoise Botterel. She's professor of medicine in Paris Créteil, and she's the president of the French Society for Medical Mycology. So Françoise is going to talk on bacterial and viral infection during cystic fibrosis and respiratory diseases. Thank you very much, Françoise.
Thank you. Good morning, everybody here and in online. I would like to thank Jean-Pierre, my friend, for giving me the opportunity to share with you some data about bacterial and viral and fungal, of course, associated respiratory fungal infection. And the question is, can we are you before dangerous lesions of best friend forever? And thank you, Dina, for your introduction about biofilms. This is my disclosure. Let's start with a few definitions. The respiratory tract infections as the number one, as you say, for consultation worldwide. Indeed, respiratory tract infections refers to a range of infection on the upper respiratory tract and the lower respiratory tract, and of course, including viruses, bacteria, and fungi, and sometimes together. We have to distinguish co-infections and super-infections. Indeed, the co-infections are concomitant infection, while super-infections are sequential infections by two different pathogens. It's very important to distinguish that. And the co-infections are associated with the severity of respiratory diseases and sometimes lead to shared resistance through biofilms. And of course, it's very important to quantify the risk of co-infections or super-infections to address the prophylactic treatment for the patients. And you can see here that the influenza virus and SARS-CoV-2 virus infect all the respiratory tree, as of course Aspergillus fumigatus. The partners of the polyinfection in the respiratory disease are, of course, all the microorganisms and the epithelial cells tract. And at the bronchial level and alveolar level. And I propose in the first part of my talk to analyze with you the interaction between microorganisms and epithelial cells, and in the second part, the interaction between microorganisms, especially in biofilms. And in fact, the co-pathogenesis of co-infections is complex and involves a dynamic interplay between the host immune defenses and the virulence of the microbes. I choose to speak about co-infections to analyze the weight of bacterial and fungal co-infections in flu. And we know that most of estimated 20 to 60 million deaths is due to bacterial superinfection rather than from direct effects of the virus. We know also that 1 in 4 patients admitted in intensive care unit with severe influenza infection presented bacterial or viral co-infection. And we know that the frequency of bacterial and fungal co-infection are highly variable from 2% to 65%. The several bacterial pneumonias following influenza with Streptococcus pneumoniae, Staphylococcus aureus, Haemophilus influenza, or Streptococcus pyogenes are recognized and are known to increase disease severity and mortality. And of course, with the recent literature about the co-infection between influenza and aspergillosis, we know that influenza virus is an independent risk factor for aspergillosis, yes, in immunocompetent and immunocompromised hosts. And we know that about 20% of influenza-infected patients are diagnosed with aspergillus. We have a lot of lessons from the literature with COVID-19, of course. There are a lot of articles from the subject, and I choose a meta-analysis published in 2021 about 118 studies with ICU and non-ICU patients. In this meta-analysis, the prevalence of bacterial co-infection after COVID-19 is about 8%, and the co-infections are higher amongst non-ICU patients. For the viral co-infection after or concomitant with COVID-19, the rate of this co-infection is about 10%, and the most virus implicated are influenza virus and respiratory syncytial virus. And for the fungal co-infections, the rate of these co-infections are about 8%, and Aspergillus is the most frequent fungi implicated. But you can see on this figure that the percentage are different between co-infections and super-infections. Now we will see together the physiological mechanism for viral infection, and we know in the flu that the virus binds to alpha-2 6-sialic acid receptor coupled to glycoproteins such as mucin in epithelial cells, and it leads a decreased mucociliary clearance. We have a loss of cilia, and you can see in the figure on the right that there is a decrease of cilia beating frequency, a dyskinesia of cilia, and we have a dysfunction of tight junctions and cytoskeleton frequently observed after viral infection. And the impact of integrity and functionality on respiratory tract contributes to creating favorable environment for secondary bacterial and/or fungal infection. For the viral and bacterial infection, after the viral infection, the window of susceptibility to bacterial co-infection is approximately to 7 days after influenza virus infection. And we have a lot of consequences after the secondary bacterial infection, after viral infection, with a lot of production of cytokines different between bacteria and virus. And it leads a cellular inflammation, a fibrosis, and apoptosis and necrosis of epithelial cells. For the viral and fungal infection, and especially for Aspergillus fumigatus, we have an alteration of respiratory tract after the viral infection. As you can see here in the figure, after infection of 24 hours. And the epithelium damage can alter, of course, the respiratory epithelium, display of transplant protein, and it's leading an exposing apical sites receptor, such as integrin alpha-1, alpha-5, and beta-1. And this exposition of apical site leads a fungal adherence in the tracheobronchial tree. After that, the damaged epithelium facilitates the adhesion of fungal conidia and, of course, subsequent invasion. And in ICU patients, the tissue repairs takes longer, and the basement membrane of the epithelial cell is more exposed, as you can see here, favoring superinfection, particularly with Aspergillus. Of course, close to the physiological mechanism, the antiviral immune response also plays a role in mixed infection. And you have here the lot of alteration of the immune response after bacterial and viral infection, with a decrease of the numbers and functions of innate and adaptive cells. After the viral and fungal infection at the alveolar level, we have a phagocytosis, fungal killings, and cytokine production impaired. The recruitment of neutrophil is also affected by both infections, and the loss of neutrophils compromise the cytokine production, the cytokine storm, which is very different between the virus and the net-mediated fungal killing. Of course, we have to release of fibrinous material which obstruct the small airways. At the end, the virus destroys the pneumocyte, and it leads to decreased oxygen and carbon dioxide diffusion capacities, and the environment changes with the hypoxic environment. We change the property of Aspergillus virulence, and we have a biofilm formation, and in the biofilm, the macroscopic morphotype and the architecture of Aspergillus fumigatus are modified because influenced by oxygen tension. Of course, all this interaction between epithelial cells and viral, bacterial, and fungal infection have consequences on diagnosis, treatment, and prevention of the poly-infection in respiratory disease. It's because in recent years, a lot of publication talk about different management and guidelines on diagnosis and treatment of this poly-infection. Let's go back on my figure with the partners of the poly-infection in respiratory disease. And now I will focus on interaction between microorganisms. And in the literature, there are few data about biofilm with viral infection. In my knowledge, I don't know. But there are a lot of few data about biofilm between filamentous fungi and bacteria. And I will take two examples to illustrate this talk. As you said, the biofilmoid is a change of life for bacteria and fungi, which change from planktonic and individual to fixed and life in communities. And the biofilm is a structured physical and metabolic community of microorganisms embedded in the extracellular, as you can see in green on the figure, which protect from external aggression and which is in contact with biotic or abiotic surface. It's very important to distinguish the bacterial biofilm and fungal biofilm because the thickness of the biofilm, fungal biofilm for the filamentous fungi is very important, and it's not the same thing with the bacterial biofilm. The biofilm have a lot of properties with localized gradients, gradients of oxygen, where the oxygen decrease according to the deepness of the biofilm. We have a gradient of nutriments, a gradient of pH, gradients of quorum something also. In biofilm, we have a lot of social interaction with a cooperation of competition between microorganisms. And of course, the problem of biofilm is the ability to survive after antimicrobial exposure. These properties include extracellular matrix that form physical basis of the biofilm, architecture and stability of the biofilm. The biofilm is characterized, I said before, by heterogeneity of gradients. Now I would like to give you two examples, the specific interaction between microorganisms. And the first one is between Aspergillus fumigatus and Stenotrophomonas maltophilia, which is a gram-negative bacteria, saprophytic bacteria, opportunistic bacteria, which is found in cystic fibrosis, in COPD, in immunocompromised with Aspergillus fumigatus. And in my lab, we analyze the cooperation between Aspergillus fumigatus and Stenotrophomonas maltophilia in mixed biofilm, and the interaction with human nasal epithelial cells, which are primary cells for the physiological relevance. Yes, in this video, you can see the formation of fungal biofilm with and without bacteria. And on the left, we can see the mixed biofilm between with Aspergillus fumigatus and Stenotrophomonas maltophilia. And you can see the slowing down of the fungus in presence in bacteria, and the typical morphology of Aspergillus fumigatus in presence with bacteria. This phenomena is called antibiosis. It's known between Staphylococcus aureus, Pseudomonas aeruginosa, and Stenotrophomonas maltophilia with Aspergillus fumigatus. And it's a molecular communication between these microorganisms known in mixed biofilm, which alter one or two partners in terms of development, morphology, or growth for one or two partners. And here, the antibiosis of Stenotrophomonas maltophilia on Aspergillus fumigatus is probably due to, in part, to secondary metabolite, as Nicolas Papon said yesterday. But here, of course, it's these secondary metabolites are secreted by Stenotrophomonas maltophilia. We have developed a lot of characteristics in our biofilm between Aspergillus and Stenotrophomonas maltophilia, and you can see from the left to the right a direct interaction between Stenotrophomonas maltophilia and Aspergillus fumigatus. Stenotrophomonas binds to Aspergillus fumigatus. You can see in orange the bacteria, and in green the filamentous fungi. We know that Aspergillus is inhibited by the Stenotrophomonas maltophilia with a specific morphology of the filamentous fungi. We observe the extracellular matrix, which signs the mixed biofilm. We observe that the increase of thickness of Aspergillus cell wall when the bacteria is present, and the last picture is on epithelial cells, and we suppose that the galactosaminogalactane, a polysaccharide of Aspergillus fumigatus, is the link between the bacteria and the fungi. This year, we analyzed too the frequency and the quality of the ciliary bead. No. Sorry for the video. And we note that when Aspergillus and fumigatus and Stenotrophomonas maltophilia are together in biofilm on the epithelial cells, we note the reductions of frequency and quality of the ciliary beads, and without no additional effect when we had Stenotrophomonas maltophilia. The same interaction have been noted by our colleague, especially Benoît Briard, between Pseudomonas aeruginosa and Aspergillus fumigatus, with first part a cooperation between both microorganisms, which facilitate the Pseudomonas and Aspergillus co-colonization when they are at distance in the epithelial cells. And after that, it seems to have a competition between both microorganisms with when two microorganisms have colonized the epithelial cell with a negative interaction with via different secondary metabolites secreted by the other microorganisms. In fact, in respiratory diseases, on epithelial cells, the microbial community is dynamic, very complex to explore, with a lot of intra and interspecies and interkingdom dialogues, as you say, Dina. There is a phenomena, different phenomena of co-occurrence, of co-exclusion between microorganisms. And we know that the iron regulation and redox potential play a key role in these interactions. For the perspective, we have a lot of interaction to explore between inter or intra-kingdom species in respiratory diseases. And for example, we analyze, thanks to the metagenomic analysis, the fungal ball due to Aspergillus fumigatus in 30 patients. And we found a specific interaction with a biofilm with Haemophilus influenza. We don't know why exactly, but we found, thanks to the metagenomic, this specific interaction between Haemophilus and Aspergillus. And we know we can discover a lot of interactions. Of course, we have also a regulation of disease exacerbation by the gut and lung axis. And we know that some forms of chronic lung disease exacerbation, such as in cystic fibrosis, in COPD, in asthma, that may be due to translocation or expansion of microorganisms content from the gut. And direct aggression to the lung could also exacerbate diseases via their effect on the lung or gut microbiota. To conclude, finally, the interaction between microorganisms in respiratory diseases, it is a dangerous lesion or best friend forever? That is the question. I think it's difficult to answer because the interaction between these microorganisms are very complex, and very complex too with host cells. And I think that nature of different pathogens, microbial and cellular context, sequence and timing of infection are essential parameters to answer these questions. I think, finally, that these microorganisms are more frenemies than enemies or friends, as described in 2019 by our colleagues Dark. I thank you for your attention.
Thank you very much, Françoise. It is really important to have a wide panel of microorganisms in mind, because of course there are many competition, cooperation, etc. So it was really interesting. So do you have one question? Yes.
What is your opinion in terms of vaccination, in respect to combined infections. Where practically could you say if someone was vaccinated that they didn't have it, or there's no influence?
I'm not. I'm an immunologist, so I cannot answer at this question. But I'm vaccinated, of course. I think for the moment we describe all the interaction between all these microorganisms. For the moment, we don't know which implication are in the context, in the medical context. We don't know. We have to explore all this interaction to answer.
Because, from gut feeling, if you have to deal with one bug, that's bad enough. But then if you have to deal with two and more, so I think my feeling is I have no data to show. I don't think there are data. Maybe there are, and I don't know. It would be worse.
Just a sort of another side of vaccinations is always that we know that over-vaccination can push people to hyperreaction and overreaction. So not that much of a problem with antiviral vaccines, but when we start talking about antifungal vaccines or antibacterial vaccines, pneumococcal vaccination, how do we actually know what we're doing?
Well, that's no problem in terms of antifungal vaccination, because there is no real vaccine here.
No, for the moment, yes.
But there are some teams that are working on Aspergillus vaccine. So, okay. So thank you very much for attending, and now we have time for lunch, and we will come back at 2 p.m. please. Thank you.